A peptide program has two halves that rarely sit in the same building. One designs the molecule, the other has to make it at scale under GMP. With an impurity profile, a regulator will accept. The company you hire for that second half is a peptide CDMO. The interesting part isn't what they do, it's what you keep and what you still have to prove. Hello, I'm Raghuvar Singh Parmar, Director Scientific Affairs at Resolve Mass Laboratories. We work with pharma and biotech teams to build the analytical data behind peptide manufacturing and tech transfer. Today, what a peptide CDMO does, how it differs from a CRO and a CMO, and which parts of the job stays yours. Before we start, I would request you one thing. If you have found anything valuable in this channel, please like and subscribe this channel. This gesture of appreciation really means a lot to us. Now let's start. Why peptide CDMOs exist now? Peptides are not new now. In 1963, Bruce Merrifield published solid phase peptide synthesis, and that one method turned peptide chemistry from a craft into a process. Approvals came slowly for decades, then the GLP-1 receptor agonist arrived, exenatide in 2005, semaglutide approved for chronic weight management in 2021, and demand for GMP peptide moved from specialist to industrial. Today, there are roughly 800 peptide programs in development, and far more sponsors need large-scale synthesis than own a plant. That gap is where the peptide CDMO lives. What a peptide CDMO actually does. So, development sitting in front of manufacturing. On the development side, route selection, solid phase, liquid phase, or a hybrid of the two protecting group strategy, coupling reagents, and a purification system that can survive being scaled. On the manufacturing side, GMP API batches, preparative HPLC purification, lyophilization, and an often sterile fill finish. Most sites also carry analytical method development and a regulatory group that writes the CMC sections describing their own process. One partner, one quality system, grams to kilograms. Now, CDMO, CMO, or CRO. Three acronyms get used as if they mean the same thing. So let's separate them. A CRO does research and testing, studies, methods, characterization data, and does not supply GMP material. A CMO manufactures to a process you already own. You hand over a defined recipe and they run it. A CDMO sits in the overlap. It builds the process, then makes the material. The difference that matters is ownership. With the CDMO, the process was developed in that building and you still have to defend it. Why peptide programs outsource? Now, the question comes here is, why not make it in-house by the sponsors? Three reasons come up in almost every program. First, capital and capacity. A GMP peptide facility is a very large build and it rarely pays back on one molecule. Second, specialist chemistry. Hindered couplings, cyclization, lipidation, and conjugation are each their own skill built over years. Third, speed. An established site already has qualified amino acid suppliers, proven purification, and an inspection history. You are buying time as much as the capacity. If this is sharpening how you'd scope your own peptide supply, a quick like helps more CMC teams find this breakdown. Now, where the chemistry gets hard. Difficulty scales here in a way that is easy to underscope. Every coupling in solid phase synthesis is a little bit below 100% efficient. Multiply that across 30 or 40 cycles and crude material carries a family of deletion and truncated sequences next to your target. Practical SPPS runs out near 50 residues and a macro cycle, a staple, a lipid chain or a payload, and you are into a much smaller pool or sites that can run it at all. This is where CDMO selection stops being commercial and becomes chemical. Now, what the impurity profile picks up. Here is the part we want to spend real time on because it's what the submission gets judged on, the impurity profile. Incomplete coupling leaves deletion and truncated sequences that elude close to the target peak. Racemization during activation gives a D-amino acid isomer with the same mass, so intact mass spectrometry alone won't flag it. Purification leaves its own fingerprint. Trifluoroacetate counterion from a preparative HPLC, residual DMF or DCM. At Resolve Mass, we sit on the analytical side of these programs. Sponsors send us peptide made anywhere in the world for independent identity, purity, and impurity work, and that data is what holds the CMC section up. Now, how an engagement actually runs. So let's talk about typical engagement. So a typical engagement runs in approximately 5 stages. Feasibility and route selection, where the site tells you whether your sequence is easy, awkward, or a research project. Process development at bench scale, analytical method transfer and qualification, so the same sample gives the same answer in 2 labs. GMP scale and validation, then release testing and the data that fills your CMC sections. Here is what does not transfer. The specification is yours, the impurity justification is yours. When a regulator asks why a new specified impurity turned up at 0.3%, the answer has to come from your file, not from a certificate of analysis. The sponsor still owns the argument. Now let's talk about key takeaways from this topic. A CDMO develops the process and makes the material. A CMO only makes it and a CRO tests it. Difficulty scales with length, cycles and payloads, not with order size. The impurity profile is where the real work shows, and the specification, the justification and file stay with you. If you are working on an IND, NDA, ANDA or a biologics program and you want an independent analytical partner alongside your peptide CDMO, identity, purity, impurity profiling and method transfer that stands on its own data, Resolve Mass Laboratory supports small and mid-sized pharma teams from first feasibility all the way through submission. Before you go, I want you to do a few things. Number 1, subscribe this channel right now. The more you subscribe, the more valuable pharma insights and expert tips and industry updates we can bring to you. Also, please comment your feedback This will really help us to improve the content, quality of the videos, and many things. And finally, share this video to at least one person who you think this would really help them. Thank you so much, and I will see you in the next time. Until then, goodbye for now.